Thank you, everyone from around the world. My name is Lorraine Rush. I'm the Vice President of Scientific Strategy and Commercial Development at Celerion. I want to share some housekeeping items with you today. Our estimated time together is 40 minutes plus time for Q&A. Please remember to submit your questions via the Q&A button, and we'll address as many of those questions as time permits. Any questions that we don't have a chance to answer, someone from Celerion will address directly within 24 to 48 hours. I'm very excited to share our insights on how to optimize renal and hepatic impairment clinical pharmacology trial designs today. Recent updates to the FDA guidances will be reviewed in line with design needs versus wants. We will also evaluate real-life case studies to support these designs, looking at the ideal patient profile which supported success. Today, we are fortunate to have Dr. Sabina Payalunga sharing her regulatory and scientific expertise to help position you, our clients, towards a successful understanding of renal and hepatic impairment study design needs in support of drug development requirements. Dr. Sabina Payalunga is the Director of Scientific Affairs at Celerion. In this key leadership position, she provides scientific, operational, and regulatory support for early clinical studies. Sabina serves as subject matter expert in metabolic diseases such as non-alcoholic steatohepatitis, NASH, and type 2 diabetes, as well as renal and hepatic impairment PK studies, and has published extensively in these areas. Sabina joined Celerion in 2015 and has progressed to become a scientific expert resource within Celerion. Sabina received her PhD from McGill University in Canada, and her training includes 3 postdoctoral fellowships. And with that, I turn it over to Sabina.
Thank you, Lorraine, for that introduction, and thanks to everyone for joining today. Celerion has over a decade of experience managing renal and hepatic impairment PK studies. Today, I'll share our insight as we unlock key study design elements for a successful renal and hepatic impairment PK study. When the liver and kidney are compromised due to impairment, drug metabolism can be altered, resulting in potential increased drug exposure and safety concerns. For new chemical entities, renal and hepatic impairment studies can help inform the label for recommendations. And renal and hepatic impairment studies can be considered as a cross between a phase 1 and a multi-site study. As a phase 1, the aspects include that it is a PK study but done in patients, so there's no therapeutic benefit. Often, a small number of subjects are enrolled. And it's a short study, normally short treatments of a single dose with intensive PK sampling. As a multi-site study, this renal and hepatic impairment study composes of enrolling 1 to 2 patients at a time. These patients are often fragile, especially the severely impaired patients. They are quite ill subjects. And because of their disease, they are on a number of medications. And this could be a major recruitment challenge. And that's where investigator feedback can be really helpful. So let's get into renal impairment PK studies. Kidney impairment can be caused by aging, chronic kidney disease, renal cancer. It's also associated with diabetes and obesity and kidney infection. And kidney damage can lead to higher drug exposure and longer half-lives, resulting in slower drug excretion. There's also the possibility that a buildup of waste products can displace drugs from binding to plasma proteins, which can affect drug clearance. And this could impact safety and efficacy, which may result in the need for labeled dose adjustments. Kidney function is calculated as glomerular filtration rate, and that's the nephron plasma volume filtration during urine formation. The direct method to measuring GFR is using inulin clearance. But this can be a complex and time-consuming method. So in a clinical setting, typically an estimation of GFR is done. And traditionally, there are two formulas which have been commonly applied. That's the Cockcroft-Gould formula and the modification of diet and renal disease formula. Both rely on serum creatinine, and creatinine is generated at a constant rate by the muscle and is completely cleared by renal excretion. The Cockcroft-Gould formula was first introduced in 1976, and this adjusts for age, weight, and gender. Later in 1999, the MDRD formula came around, and this additionally adjusts for race, ethnicity, as well as normalize for body surface area. There are limitations with these formulas. For Cockcroft-Gould, it was actually developed in a quite a small sample size, and it has been validated since. But because the assay was developed in '76, this is no longer available. And it's estimated that it likely is 10 to 20 times higher than the current methods. So because of this, the formula can actually overestimate and incorrectly classify severe patients as moderates. And it is better at estimating healthy subjects, and that's why it is preferred for PK studies for healthy subjects. On the other hand, MDRD does use a standardized serum creatinine assay, and this is commonly used in clinical practice. The guidance actually recommends this for renal impaired subjects. Newer formulas include CKD-EPI. It has results that are similar to MDRD and is recommended by kidney organizations, though not currently endorsed by any regulatory agencies for PK studies. And then as an alternative for classifying healthy subjects, 24-hour urine creatinine is also available, though this is an intensive time-consuming method. The FDA recommends that a dedicated renal impairment PK study be performed for drugs that are intended for patients with kidney disease and that are primarily eliminated through the kidneys. And that's defined when the fraction excretion of the unchanged drug is over 30%. It's also recommended for therapeutic proteins with a molecular weight below 69 kilodaltons. This study is not needed for volatile or topical drugs if the drug will be a single dose administration or therapeutic proteins with a high molecular weight. Last month in September, the FDA released new guidance for renal impairment studies. And the new guidance now actually addresses the definition of kidney failure. It gives input into full and reduced study design, as well as the requirement for sample size justification and data modeling. I'll go through some of these changes within this webinar. A full study is recommended when the drug is intended for patients with kidney disease and is mainly eliminated by the kidneys. This study explores the full spectrum of renal function with cohorts ranging from normal to mild, moderate, severe, and kidney failure. Now, the new guidance actually provides us with a definition of kidney failure as a GFR less than 15 or patients on dialysis during their non-dialysis day. This is a change from the previous guidance, which referred to this class as end-stage renal disease. and the term has been updated to be consistent with clinical practice guidelines. An exciting new option for a full study is the use of a compressed design. So if a new chemical entity does have a wide therapeutic range, they may opt for this compressed design, which reduces the number of cohorts from 5 to 3. For a reduced study, this explores both ends of the kidney function spectrum, and it looks at normal compared to severe renal impairment. It's an option when the drug is not predominantly eliminated by the kidneys. Now, this study design has changed quite a bit from the previous guidance. In 2010, when the guidance was released, it actually recommended that end-stage renal disease patients not yet on dialysis be used for a reduced study. But after discussion with an advisory committee, the FDA agreed that this population would be rather difficult to recruit. So they suggested that the GFR less than 30 would be an appropriate cohort. Now, the updated guidance goes one step further and recommends a severe group, which has a range of 15 to 29. This does omit the option to enroll patients that are less than 15, so it may have impact on recruitment and study design moving forward. For a reduced study, there is no difference in PK, then no further action is needed. If there is a finding, the other stages should all be explored. I'd like to go through a case study example, and this is a study that we did a couple years ago. It's a full study that was 2 parts. In part 1, we looked at mild and moderate patients, and part 2, severe. Now, back then, there was no sample size requirement in the guidance, and typically we opted for 6 to 8 subjects per cohort. Data from previous PK informed us that there was quite high variation, so we recommended at least 8 subjects and 3 sites for this study. The sponsor, however, opted to go for 6 patients per group just because they were trying to minimize time and cost. During the Data Safety Monitoring Board review of the data from the mild and moderate patients, they did see that there was quite some high variation. So the protocol had to be amended and the cohort sizes were increased to 10. Even with increasing the cohort size, we were able to complete conduct within 16 weeks. And once all the data was put together, there was no difference between the mild or moderate patients in the control group. So this is an example that highlights the appropriate need for a substantial sample size. And the FDA actually updated the guidance to now include and make a recommendation for sample size justification. So if the inter-subject variation is greater than 50%, the sample size could exceed 14 patients. This is almost twice the number of patients that were traditionally enrolled. Now, we will have more confidence in the outcome and the data, but it will have a dramatic effect on timelines, number of studies, sites, as well as study costs. Our biostatistician actually calculated recommended sample size to achieve 80% power based on different standard deviations and variations. And you can see in the table, the N number ranges from 7 to 24. So this change to the guideline, I think, will have most impact on future studies. For study design, there are several options. There is a parallel design, and that enrolls each renal function category with controls at the same time. The advantage of this is that you can potentially reduce duration of the study overall. Then there's the adaptive design. This is a two-part study where your patients from one category are recruited in part 1, and then the next category of patients are recruited in part 2 with their matched controls. An advantage of going with this model is that part 2 may not be required. So this would kind of resemble that reduced model and can lead to potential cost savings. Then there's a sequential study design. It's a 2-period study. So this is ideal for looking at patients on or off hemodialysis, or 2 cohorts to look at dose exploration during hemodialysis. For drug administration, the drug could be dosed but in a single dose study or a multiple dose study. The single dose is typically used, and that's when the PK is dose proportional and time independent. But if PK shows that the drug is dose or time dependent, then multiple doses may be needed. And here we can consider a loading dose to achieve steady state a little quicker. For sample collection, it's important to collect total and unbound drug concentrations as well as urine. If plasma proteins are less than 80%, then typically total drug concentrations would be fine. If plasma protein binding is more than 80% and it is concentration dependent or affected by metabolites or time dependent, then it's important to measure the unbound drug concentration for each sample. If that's not the case, then we can just look at unbound drug concentration for one sample per patient. Here's another case study example from a previous study that we did. This is a study in which the previous PK informed us that steady state was achieved in about 2 weeks, and there were 3 metabolites. The sponsor wanted to do a reduced study design, and we're looking at 10 severe subjects and 10 controls. Because of the long steady state, we then went for the loading dose option, followed by 20 days of daily dosing, but the drug was dosed at a lower concentration. Our strategic approach was to use 2 sites, and all the conduct was completed within 20 weeks. For the study findings, we did see that there was an increased exposure in the AUC of around 25% in the active drug, and the metabolites showed about 40% increase in exposure. So this study then signaled that there would be need to explore the full spectrum of renal function. Renal impaired patients often have several comorbidities, such as cardiovascular disease, diabetes, obesity, and hypertension. So it's important to have inclusion criteria that are practical. And we worked with our PIs to come up with a list of recommended inclusion criteria that we found works best. The first is to consider increasing the age up to 85 years. Now, a lot of sponsors might be hesitant to go that high, but it does really increase the patient pool. And the sites do have appropriately matched controls. Another key inclusion criteria is to have BMI up to 40. Again, diabetes and obesity are risk factors for this disease and associated with renal impairment. So having a wide BMI also increases the patient pool. Hemoglobin levels at 9 or more could be considered for shorter PK studies with minimum blood volume. And then other considerations include allowing for repeat labs, as well as having no stability requirement, or if needed, setting that to 25%. We also suggest no ECG requirement unless there is a QTC signal. And then, if that's the case, have it at less than 500. I'd like to share an example of a recent study that we completed. And this is a study that really had optimal inclusion criteria. It's a two-part stage study that enrolled part 1, mild and moderate patients, and then part 2, severe patients. The previous PK informed us that the half-life was about 2.5 hours. So it's a relatively short study. We followed these patients out for 24 hours. We worked with our PIs and sponsors to develop optimal inclusion criteria, which included a BMI up to 40. For this study, moderate smokers were allowed, repeat testing was allowed, and renal stability was at the discretion of the PI. There was also no criteria for ECG. Our approach for this study was to go with 2 sites, and we forecasted conduct would be completed within 16 weeks. But actually, we finished conduct 1 month ahead of schedule in 12 weeks. And this is due to the optimal IE criteria and excellent project management. For healthy controls, they should be matched based on age, sex, body weight, and in some cases, race and ethnicity. We typically recommend age be plus or minus 10 years, and the BMI window be plus or minus 5 to 20%. For both patients and controls, it's important to enroll the reflected demographic of the intended population. So that means that if the drug is going to be intended for an Alzheimer's indication, for example, then you'll want to consider enrolling elderly patients as well as elderly healthy controls. It's important to note that GFR does decline with age. So the healthy control range, which is typically 90 or more, should potentially be reduced by 10 points and be, for example, more than 80 in this situation. Now, controls can be matched individually, 1-to-1, or mean match, and each system has its pros and cons. For 1-to-1 matching, here we can enroll patients and controls at the same time. But it could be challenging if a patient has an uncommon characteristic, trying to find that 1-to-1 match. For mean matching, you require fewer controls, but you have to wait until all of your patients are enrolled first. Now, it's good practice to enroll healthy subjects at the same site as the patients, so they're exposed to the same environment and study processes. For a dialysis study, this is needed when the drug is intended for kidney failure patients undergoing dialysis. It's not necessary if the drug has a high molecular weight, which is unlikely to be cleared by the dialysis process, or has high plasma protein binding. There are 3 key questions a dialysis study should address, and they are: is dose adjustment needed due to the dialysis? If so, how much? And when relative to dialysis? An additional advantage of this study is that it can provide insight into overdose treatment. In the US, high-flux intermittent hemodialysis is most common and typically recommended for PK studies. Now, the dialysis study will actually look at two PK profiles. One, when the patient is off their dialysis, to look at the effect of severe impairment on PK. And the second is when they are on dialysis during the dialysis procedure to look at the impact of hemodialysis on drug clearance. Key design considerations for this type of study is to commence hemodialysis just before the anticipated Tmax. Also, it's important to collect blood samples before and during the dialysis period, as well as samples for plasma protein binding before and after dialysis starts. In addition, you want to be able to capture key dialysis parameters as well. Here's an example of a hemodialysis study that we ran. It's a two-part study. On day 1 of period 1, the subjects received a single dose of the drug just after their regular scheduled hemodialysis. And then on day 1 of period 2, they received a dose 2 hours prior to their hemodialysis. Pre and post dialyzer plasma was also measured. For this study, we went with one site, and conduct was completed in 8 weeks. During the washout period, which was about 7 days, we did offer the patients housing to ease any transportation burden. For studying findings, we found that both the AUC and the Cmax were lowered by 20% when the patients were undergoing hemodialysis. So that indicated that this drug would not be recommended during hemodialysis and should be dosed on their non-dialysis days or following hemodialysis. Next, we'll get into hepatic impairment PK studies. Hepatic impairment can be caused by viral hepatitis, cirrhosis attributed to alcoholic or non-alcoholic fatty liver disease. It's also associated with bile duct disease, hepatic carcinoma, and genetic diseases like alpha-1 antitrypsin. Liver damage can also be caused by hepatotoxicity caused by drugs. Liver damage can reduce first-pass clearance, resulting in longer half-life and higher drug exposure. It can also reduce the synthesis of albumin and plasma protein, which then can lead to an effect on drug distribution and drug clearance. This can impact safety and efficacy, and therefore may require a label dose adjustment. A dedicated hepatic impairment PK study is recommended when the drug is intended for patients with liver dysfunction and metabolism accounts for roughly more than 20% of the absorbed drug. It's also needed if the drug has a narrow therapeutic range. Hepatic impairment PK study is not needed if the drug is volatile, if it's cleared entirely by the kidneys, intended for single dose administration only, or has a wide therapeutic range with minimal hepatic elimination. For study design, there is also a full and reduced study option. Here, a full study would have 3 patient groups, the mild, moderate, and severe, compared to a control. And the guidance does suggest at least 6 patients per cohort. In a reduced design, the 2 cohorts include moderate patients and controls. Now, any findings that come from the moderate category would then apply also to mild patients. And for labeling purposes, the label would just say that the severe category would be contraindicated because it was not evaluated. A reduced study, the guidance then suggests about 8 patients per cohort. We have similar design options such as single or multi-dose, parallel and adaptive, and then individual or mean matched controls, as I previously described. Now, for the guidance, hepatic impairment is classified as cirrhosis severity using the Child-Pugh system. This is a score that looks at both clinical findings and laboratory results. So it evaluates encephalopathy, ascites, as well as bilirubin, albumin, and prothrombin time, and totals up the scores for each finding. Mild liver impairment is class A with 5 to 6 points. Moderate impairment is class B, and severe impairment is considered class C. Many studies will enroll patients with any hepatic disease as long as cirrhosis was confirmed by biopsy or a physical examination, liver imaging, or endoscopy findings. But over the past couple of years, there's been a surge in non-alcoholic steatohepatitis drug development, and guidance for NASH drugs actually came out in 2018. Here, the guidance recommends that NASH patients be enrolled for hepatic impairment PK studies. And this was reinforced during a hepatic workshop that the FDA sponsored last month. Hepatic impairment patients also have several comorbidities and are on multiple medications. So it might be important to withhold certain medications if there is anticipated drug interactions with known inhibitors. And the PIs and protocol writers can help determine what should be withheld and what should be contraindicated. These patients are notoriously clinically unstable as well. So mild patients, they can often shift from screening to dosing day. Moderate patients that are borderline can even switch between classes. And the severe patients, well, they're the hardest to recruit, and they tend to drive enrollment and site numbers. Again, based on input from our PIs, here's our optimal inclusion-exclusion criteria. We recommend that the age be up to 75 and BMI up to 40. If CYP-A1 is not involved in the drug's metabolism, then light smokers should be considered. Because these patients have liver dysfunction, it is expected that their liver function tests will be abnormal. And they also have some form of kidney disease as well. So having a GFR of 50 or more is optimal. Again, we recommend repeat labs when possible, as well as allowances for concomitant medication related to their diabetes, hypertension, or thyroid disease. And similarly, considerations for anemia and no ECG criteria unless necessary. Here's an example of hepatic impairment PK study that we did. It's a two-part study, and a single dose adaptive design. In part one, we looked at moderate impairment as well as controls. And in part two was mild impaired patients and their matched controls. For this study, we opted for 2 sites and completed conduct of part 1 in 8 weeks. The results showed that there was a 2-fold increase in the area under the curve and about a 60% increase higher Cmax in the moderate group compared to their controls. So because there was such an obvious effect here on the PK, they did not proceed with part 2 of the study, really using that reduced design. And this allowed for potential savings of time and money. In these last few minutes, I'd like to spend a bit of time going over study management and our study sites. At Celerion, we work with a network of clinical pharmacology sites that specialize in renal and hepatic patient populations. We have a really long history of partnering with these sites and keep adding new sites to our network. Our clinical program managers are experts in managing multi-site studies. And we've recently introduced VivaVault as our management program platform to streamline workflow. We also provide complete solution and supportive services for any PK patient study. There are several factors that will contribute to the number of sites we propose, and that includes speed. We want to know when the data is needed for the new drug application. That will depend on how many sites we will suggest. Another big factor is difficult to recruit inclusion-exclusion criteria. And we'll work with the sponsor and the PIs to help develop open and flexible criteria to find that optimal criteria for each study. With the new renal impairment PK guidance that came out, the number of mild and moderate patients needed will also likely impact the number of sites we suggest, especially if a study needs up to 14 or 16 patients per cohort. And then severe patients. They've always been the main driver of enrollment rates and the number of sites. Now, COVID is also another consideration for the number of sites needed. I'm happy to report that all the sites we work with are open and they have several COVID-19 measures in place. The patients and participants, as well as staff, they're all in full PPE. The patients are questioned about their symptoms before and at the study visits, and they are kept separate and individually confined as well. We are seeing a trend that COVID is increasing enrollment time for each severe patient, and we think that this might be related to some hesitancy of these participants from actually doing a study due to increased risk of exposure to the virus. In summary, the optimal study design for a renal, hepatic, or PK study will depend on 6 key factors. Drug elimination. How is the drug eliminated and what is the previous PK information? Timing is another factor. Timing will influence the number of sites proposed, as well be affected by the severe inpatient enrollment. Expertise. Our expert PIs consult early on inclusion-exclusion criteria, as well as any potential safety issues to mitigate barriers to enrollment. ConMeds is another big one. The protocol review team should carefully review for any potential interactions and recommend withholding certain medications if necessary. For healthy controls, we can recommend mean or individual matching for potential time and cost savings. And finally, excellent study management. With dedicated clinical program managers and CRAs located nearby, we can provide excellent study management. I'd like to thank you for your attention and now pass this back to Lorraine. Thank you.
All right, that was really very nice. Thank you so much, Sabina. And with that, I think we'll move into the question and answer session. And we already have a few questions popping up, so I'd like to go ahead and look through those. The first question for Sabina is, what approaches are available if a sponsor is seeking a hepatic or renal PK waiver?
Well, that's a great question. And there are a couple options. There's population PK analysis, and that's measuring the and capturing drug information during phase 2 and phase 3 in patients that actually have renal impairment. And it's important to have sufficient number of patients over a range of renal function classes. Also, if the unbound drug concentration needs to be measured, that appropriate steps are taken to capture that. You also want to make sure that renal function is estimated using the same formula across each site. And then there's also physiologically based modeling. And this is new to the renal impairment guidance. It can be used to show that there is likely no change or meaningful change with renal function, and could be a rationale not to perform a study in renally impaired patients. PBPK can also be used to support information regarding renally impaired studies.
Well, it sounds like there's quite a spectrum of considerations to work through. So that's very helpful and informative. The second question is, what happens if toxicities occur during a renal or hepatic impairment study? Should the study continue?
That's an important question, and toxicities can occur. They have occurred. So the protocol should identify a process of really determining when toxicities occur, and that should be pretty well laid out in the protocol. And when they do occur, the regulators don't expect you to continue dosing patients. In this case, proper documentation is important, and then you could state that certain groups were not studied because of potential toxicities, and this group is not recommended, or it's contradictory for a specific patient group.
Okay. Question number 3, are there any requirements for measuring or processing unbound drug concentration?
Yeah, so there's two types of modalities that we could use to measure unbound drug concentration, typically for these studies. One is ultrafiltration, and the other is equilibrium dialysis. For ultrafiltration, can be done at most sites, and the sites we work with have this capability. They use ultrafiltration cones, and they have the centrifuges in place, able to work with fresh blood samples. Often 1 to 2 mils of blood is required for this type of procedure. So you need to make sure that your protocol accounts for this, and the IRB may request minimum volumes be used for these patients as well.
Okay, and then the final question, do EMA guidelines differ from FDA guidance?
Good question. So typically the two sets of regulatory agents, they tend to have similar guidances. A few key minor points: the EMA for renal impairment studies, they actually stipulate a sample size of 6 to 8 patients. And they also recommend that a defined renal impairment method be used, such as inulin clearance, which is an exogenous method, rather than the estimation to appropriately define these patients. And the EMA guidance also seems to lean or prefer the CG method of reporting in addition to inulin clearance over MDRD. For hepatic impairment PK studies, the two guidances are pretty similar. Only one point here is that thrombin time should be reported in seconds rather than INR.
Understood. All right. Well, thank you so much, Sabina. And thank you, everyone, as this concludes our webinar. A copy of the webinar will be available on our website. And as a reminder, Celerion will address all the remaining questions directly within 24 to 48 hours. And again, thank you very much.